Hey, sleepers. Tonight, we're learning a secret about the brain. Imagine a vast, quiet city at night. The avenues are dark, but billions of tiny lights flicker, sending messages from one district to another. This is your brain, a universe of thought and feeling, humming with silent electric life. And tonight, we are going to find a hidden power in that city. A cell with a secret identity, a spark of renewal that exists even when some of the lights go out. So before you get comfortable, take a moment to like the video and subscribe. It helps our little corner of the internet grow, and it ensures you won't miss our next journey together. And while you're there, feel free to leave a comment. Tell us where you're listening from and what time it is in your part of the world. It's always so wonderful to think of us all, scattered across the globe, settling down together to explore a quiet mystery. So close your eyes, take a deep breath, and get ready to drift off as we explore the brain cell that regrows after death. Let us begin our story, not in the bustling, modern laboratories of today, but in a quieter, more candlelit time. Let's travel back to the late 19th and early 20th centuries. The world was changing fast. The first automobiles were beginning to chug along cobblestone streets, and electric lights were starting to push back the night in major cities. In the world of science, a revolution was underway. Scientists were peering through brass and glass microscopes, discovering a world of cells, of bacteria, of the very building blocks of life. And in this world, there was a giant. His name was Santiago Ramon y Cajal. Cajal was not just a scientist. He was an artist, a man of immense passion and meticulous detail. He was born in a small town in Spain, and he had a rebellious, artistic spirit from a young age. His father, a doctor, pushed him toward medicine, but Cajal's heart was always with the visual, with drawing what he saw. This unique combination of scientific rigor and artistic sensibility made him the perfect person to map the brain. At the time, the brain was a complete mystery. Scientists looked at brain tissue under the microscope, and it looked like a tangled, messy jungle, a chaotic web of fibers. They called it a reticulum, a single continuous network, like a spider's web where every thread was connected to every other thread. But Cajal, using a special staining technique developed by his rival, Camillo Golgi, saw something different. He saw that the brain was not one continuous web. It was made of individual cells, separate, distinct units. He called them neurons. He drew them with breathtaking beauty and accuracy. His drawings looked like ancient, intricate trees, with long, sprawling roots and delicate, branching crowns. He showed how these tree-like cells were positioned next to each other, close enough to pass messages, but not physically fused together. This was the neuron doctrine, the fundamental idea that the brain is made of individual cells, and it won him the Nobel Prize. It is the foundation of all modern neuroscience. But in his meticulous study of this intricate forest of the mind, Cajal came to a conclusion that was as influential as it was stark. He looked at the adult brain, the brain of a mammal, of a human, and he saw no new growth. He saw that when a neuron, one of his beautiful cellular trees, was damaged or died, it was not replaced. The forest did not regrow. In his own words, he issued what became known as the harsh decree. He wrote, "In the adult centers, the nerve paths are something fixed, ended, and immutable. Everything may die. Nothing may be regenerated." For over a century, this was the central truth of neuroscience. The brain was seen as a magnificent but fragile machine. You are born with all the neurons you will ever have. As you learn and grow, you strengthen the connections between them. You prune away the ones you don't need, but you don't get new ones. If you had a brain injury, a stroke, or a disease that killed neurons, that was it. The loss was permanent. The brain was a fixed, non-regenerative organ. Its structure was permanent. Think of it like a sculpture carved from a single precious block of marble. An artist can spend a lifetime carving it, creating a masterpiece of incredible complexity. But if a piece chips off, if an arm breaks, the marble cannot grow back. The damage is forever. You can try to glue it back on, but it will never be the same. This was the view of the brain. A beautiful, finite, and ultimately fragile masterpiece. This idea seemed to be confirmed by what doctors saw every day. A person who had a stroke in the part of the brain that controls movement might lose the use of their arm forever. Someone with damage to their memory centers would struggle with memory for the rest of their life. The evidence seemed to be all around us. The dogma of the static, unchanging brain was set in stone. It was a sobering thought, a reminder of our own fragility. It shaped medicine, it shaped our understanding of aging, and it shaped our very idea of what the self is. If our brain is who we are, and our brain cannot heal itself, then who are we after it has been broken? It was a heavy thought, a truth that seemed as solid and unyielding as granite. But as we will see, even granite can have hidden cracks, hidden potentials, waiting for the right conditions to be revealed. The story of the brain's permanence, it turns out, was only the first chapter. And a new character was waiting in the wings, a humble, overlooked cell that was about to change everything. For all the attention lavished on the neuron, with its flashy electrical signals and its starring role in thought and consciousness, it is not the most common cell in your brain, not by a long shot. In fact, neurons are vastly outnumbered by another class of cells, a diverse and busy population known collectively as glial cells. For a long time, these glial cells were the unsung heroes of the nervous system. The very name glia comes from the Greek word for glue. Scientists like the German pathologist Rudolf Virchow, who first described them in the mid-1800s, thought their main job was just to hold the important neurons in place. They were considered the packing peanuts of the brain, the structural support, the brain glue. Important, yes, but passive. Boring even. Among this diverse family of glial cells, one type is the most abundant of all. It is a cell with a beautiful name that matches its shape, the astrocyte. Astro means star, and cyte means cell, the star cell. And if you look at one under a microscope, you can see why. It has a central body with numerous fine, branching processes that radiate outwards in all directions, like the rays of a star. They are beautiful, intricate, and they are everywhere in your brain and spinal cord, nestled in between the neurons, wrapping their delicate arms around them. For decades, we thought we knew the astrocyte's job description. It was the neuron's loyal support staff, a tireless housekeeper, a dedicated bodyguard, and a meticulous gardener, all rolled into one. Let's take a moment to appreciate these traditional roles, because they are vital. Without the astrocyte, the neuron could not function. First, imagine the astrocyte as the brain's master gardener. Neurons are incredibly high-energy cells. They are constantly firing, constantly working. They need a steady supply of fuel, of energy, to do their job. Astrocytes help with this. They have little feet that connect directly to the brain's tiny blood vessels, the capillaries. They can draw nutrients, like glucose, from the blood, but they don't just hand it over to the neuron. They process it first, breaking it down into a more readily usable form of energy, called lactate. They then pass this specialized fuel directly to the neurons they care for. It's like a personal chef preparing a custom meal for a star athlete. They are tending to the garden of the mind, making sure every plant has the food it needs to thrive. Next, picture the astrocyte as the brain's diligent janitorial crew. When neurons fire, they release chemicals into the small spaces between them. These chemicals are called neurotransmitters. They are the messages. But after the message has been delivered, those chemicals can't just be left floating around. It would be like leaving letters scattered all over the street after the mail has been delivered. It would create noise and confusion. Astrocytes are there to clean it up. They have specialized pumps on their surface that actively suck up the excess neurotransmitters like tiny vacuum cleaners. They also clean up other waste products that cells produce, keeping the environment around the neuron pristine and perfectly balanced. A clean workspace is a productive workspace, and astrocytes are the best janitors in the business. Finally, think of the astrocyte as the brain's most exclusive security guard. They are a key component of something called the blood-brain barrier. This is a highly selective border that separates the blood circulating in your body from the fluid in your brain. Your blood can contain all sorts of things: hormones, immune cells, stray chemicals from the food you ate. Many of these would be disruptive or even toxic to the delicate environment of the brain. The astrocytes use their star-like arms to wrap around the blood vessels in the brain, reinforcing this barrier. They act as bouncers at the door of a very exclusive club, deciding what gets in and what stays out, protecting the neurons from potential harm. So, for a very long time, this was our picture of the astrocyte. The gardener, the janitor, the guard. Essential, yes. Crucial for keeping the brain healthy, absolutely. But they were always in the background. They were the supporting cast. The neurons were the stars of the show, processing information, creating memories, generating our thoughts. The astrocytes were just the glue, the staff, the framework. They were not thought to be capable of processing information themselves, and they were certainly not considered capable of becoming anything else. They were astrocytes, and astrocytes they would remain. A completely different type of cell from a neuron, with a different job, a different identity, a different destiny. But science is a story of surprises. It's a story of looking closer at something you thought you understood and realizing you've missed the most incredible part. Researchers started to notice that astrocytes were more involved than they thought. They found that astrocytes could listen in on the conversations between neurons. They even seemed to be able to talk back, releasing their own chemical signals that could influence how the neurons fired. They were not just passive support. They were active participants in the brain's network. The brain glue was starting to look a lot more interesting. The supporting actor was stepping into the spotlight, and it was about to reveal a secret talent that would challenge the very foundations of neuroscience, a talent for transformation that no one, not even Kyle, could have ever imagined. This is where our story takes a turn from the established and the understood into the realm of the truly revolutionary. It's a story of cellular shapeshifting, of a cell being given a new identity, a new purpose. The scientific term for this process is transdifferentiation. It's a long word, but the idea behind it is beautifully simple. Think of it like this. In your body, most cells are specialized. A skin cell is a skin cell. A muscle cell is a muscle cell. They have their career path chosen for them, and they stick to it. Transdifferentiation is like a cell undergoing a complete career change at a fundamental level. It's like a baker deciding to become a blacksmith. They don't just learn a new skill. They transform their entire workshop, their tools, their very nature to fit the new role. For decades, it was thought that once a cell like an astrocyte was differentiated, once it had become a mature astrocyte, that was it. Its fate was sealed. But a new generation of scientists began to ask a radical question. What if it wasn't? What if you could persuade a cell to change its mind? The key to this persuasion lies deep within the cell, in its nucleus. The nucleus is the cell's control center, and inside it is the DNA. The DNA is like a massive library, containing the instruction manual for building the entire body. Every single cell in your body has the same library, the same complete set of blueprints. A skin cell has the blueprint for how to be a neuron. A neuron has the blueprint for how to be a liver cell. The difference between them is which blueprints they are reading. The librarians that control which books are open and which are kept closed on the shelf are special proteins called transcription factors. These transcription factors are the master switches. They bind to the D and IN turn certain genes on or off. An astrocyte has transcription factors that keep the astrocyte chapter of the library open and the neuron chapter firmly shut. The revolutionary idea was this. What if you could sneak a new librarian into the cell? A librarian that would march into the nucleus, close the astrocyte book, and open the neuron book instead. This is precisely what scientists, like Dr. Magdalena Gutti in Germany, a true pioneer in this field, and Dr. Gong Shun, then at Penn State University, began to achieve in the early 2000s. They identified specific transcription factors, proteins with names like NURD1, SOX2, and ASCL1. These are proteins that are normally active when neurons are first being born in the developing brain. They are the master regulators of the becoming a neuron program. The challenge was how to get these proteins, these new instructions, inside a mature astrocyte in a living brain. They used a clever delivery system, a harmless, disabled virus. Think of the virus as a tiny biological envelope. The scientists removed all the harmful viral parts and replaced them with a genetic code for one of these key transcription factors. They could then inject these viral envelopes into a specific area of the brain. The envelope would deliver its message to the nearby cells, including the abundant astrocytes. And then something incredible happened. The astrocytes that received this new genetic message began to change. It wasn't instantaneous. It was a slow, deliberate transformation. The cell would first begin to shut down its astrocyte identity. It would stop producing the proteins that made it an astrocyte. Its star-like shape would begin to soften and change. Then, guided by the new transcription factors, it would start reading the neuron chapter of its DNA library. It would begin to grow the long, wire-like projections of a neuron, an axon to send signals, and branching dendrites to receive them. Its internal machinery would change. It would start producing the proteins needed to make neurotransmitters, the chemical messengers of the brain. Within a few weeks, a cell that was once a humble astrocyte, a gardener and a janitor, had physically and functionally become a neuron. Scientists could see it under the microscope. Its shape was that of a neuron, and using delicate electrodes, they could detect electrical signals. It was firing. It was talking. It was participating in the neural conversation. Even more remarkably, these new astrocyte-derived neurons were forming connections, or synapses, with the original neurons around them. They were integrating themselves into the existing network. They were not just looking like neurons. They were acting like them. This was not a small discovery. It was an earthquake. It directly contradicted Kyle's century-old decree. Here was a way to create new neurons in the adult brain, not from a special population of stem cells, but from the most common cell already there. The brain, it turned out, contained the seeds of its own regeneration, hidden in plain sight within its own support staff. The brain glue was not glue at all. It was a secret reserve of potential neurons, a dormant workforce waiting for a new set of instructions. The idea that brain damage was permanent suddenly had a challenger. The marble sculpture, it seemed, wasn't made of marble after all. It was made of something more like clay, something that, with the right touch, could be reshaped and repaired. The implications were staggering, and the story was about to get even more mind-bending. Now, we arrive at the part of our story that sounds like it's been lifted from science fiction. It's the core twist, the discovery that is so profound it forces us to reconsider the very boundary between life and death at a cellular level. The ability of astrocytes to transform into neurons was already a monumental discovery. It offered hope, a new path forward. But scientists, in their endless curiosity, pushed the question one step further. They knew this potential for transformation existed in a living brain. But how resilient was this potential? How long did it last? Does all cellular potential, all possibility for change, simply vanish the instant that life ceases? To answer this, researchers embarked on a series of truly remarkable experiments. These studies are conducted with the utmost respect and care, using brain tissue that has been generously donated to science by individuals and their families. It is a profound gift, allowing knowledge to blossom even after a life has ended. In these studies, scientists obtained small samples of human brain tissue, not from a living patient, but from post-mortem donors. These samples were collected hours, and in some cases, even a day or two, after clinical death had occurred. Let's just pause and visualize this for a moment. Picture a laboratory, quiet and sterile. A small piece of human brain tissue rests in a petri dish, in a carefully prepared nutrient broth that mimics the environment of the brain. The person this tissue belonged to is gone. Their consciousness, their memories, their life has ended. By all conventional measures, the processes of life in that brain have stopped. The vibrant, crackling city of the mind has gone dark and silent. Or has it? The scientists took these post-mortem tissue samples and performed the same procedure we discussed before. They introduced the special transcription factors, the molecular keys like Neurod1, into the cells within this tissue. They were essentially sending a new set of instructions into a system that was, for all intents and purposes, shut down. And then they waited. And they watched. What they saw was astonishing. Even hours after death, a spark of potential remained. The cells in this tissue were not just inert, decaying matter. They were still, in a very real sense, responsive. And specifically, the glial cells, including the astrocytes, were still listening. Upon receiving the new instructions, these astrocytes from a brain that had ceased to live began to transform. They started to shed their glial identity. They started to express neuronal genes. They began to change their shape, growing the characteristic axons and dendrites of a young neuron. This discovery is deeply profound. It tells us that the cellular machinery for change, the fundamental plasticity of these cells, is incredibly robust. It can outlast the life of the organism itself. The library of blueprints inside the astrocyte nucleus doesn't just slam shut at the moment of death. It remains accessible. The cell's ability to read a new chapter, to embark on a new path, persists in this quiet twilight period. It challenges our black and white view of life and death. We tend to think of it as a switch, on, then off. But biology is rarely that simple. It's more like a complex orchestra playing a grand symphony. When the conductor leaves the stage, the music doesn't just stop instantly. The notes hang in the air. The instruments slowly cool. The musicians pack up their things. There is a process of fading away. And what this research shows is that even as the symphony of consciousness fades, some of the musicians, the astrocytes, are still sitting in their chairs, instruments in hand, capable of playing a new tune if a new sheet of music is placed in front of them. This has very little to do with bringing a person back to life, of course, but it has everything to do with understanding the fundamental resilience of our own biology. It reveals a hidden layer of potential that we never knew existed. It's a humbling discovery. It shows that even in tissue we once would have considered inert, there is a latent capacity for renewal, a cellular echo of life. Think of an ancient seed found in a tomb after thousands of years. It looks like a dead, stony pebble. But if you give it the right soil, the right water, the right light, it can still sprout. The potential for life was dormant inside it all along, waiting. The astrocytes in the postmortem brain are like those seeds. They hold a dormant potential for a new identity, a potential that is so strong it can even survive the death of the brain that houses them. This incredible resilience is not just a scientific curiosity. It is a source of immense hope because it suggests that the astrocytes in a brain that is damaged, but still very much alive, are more than ready for a second chance. And this brings us to the very practical and very hopeful applications of this amazing discovery. To understand how this remarkable cellular shapeshifting could be used as a therapy, we first need to understand what the brain does when it gets hurt. When there is an injury, like a stroke, which is when blood flow is cut off to a part of the brain, or a traumatic brain injury from an accident, the brain's emergency response systems kick into high gear. And at the very center of this response, we find our star cell, the astrocyte. Immediately following an injury, the astrocytes in and around the damaged area change their behavior dramatically. They enter a state known as reactive astrogliosis. They become reactive astrocytes. Imagine a quiet neighborhood where a fire has just broken out. Suddenly, the whole community is in a state of alarm. People are running around, shouting, putting up barriers. It's chaotic, but it's a response to a crisis. This is what happens with reactive astrocytes. They swell in size. They multiply. They interlock their starry arms with each other, forming a dense, thick barrier around the site of the injury. This barrier is called a glial scar. For a very long time, the glial scar was seen as one of the main villains in the story of brain injury, and in many ways, it is. The scar is like a hastily built concrete wall around the disaster zone. It does have a useful short-term purpose. It walls off the damaged area, containing the inflammation and preventing toxic chemicals from the dying cells from spreading to healthy, neighboring brain tissue. It's a form of damage control. But this wall, this glial scar, is a major problem for long-term recovery. It creates a physical and chemical barrier that prevents new connections from being made. Imagine a healthy neuron from a nearby area trying to grow a new axon, a new connection, to help rewire the damaged circuit. When that growing axon reaches the glial scar, it stops. It can't get through. The scar releases chemicals that actively repel new neuronal growth. So, while the scar helps to contain the immediate damage, it also prevents the brain from using its natural, albeit limited, ability to rewire and heal itself. It's a permanent roadblock on the path to recovery. This is why brain injuries so often lead to permanent deficits. The glial scar, formed by those reactive astrocytes, stands in the way. It's a classic example of a biological response that is helpful in the short term, but harmful in the long term. A double-edged sword. But here is where our new understanding of astrocytes changes the story completely. We used to look at this glial scar and see only a problem, a barrier to be broken down, dissolved, or somehow bypassed. But with the knowledge of transdifferentiation, we can now look at that very same scar and see an incredible opportunity. Think about it. Who forms the glial scar? The reactive astrocytes. Where is the glial scar located? Precisely at the site of the injury, right where new neurons are needed most. What is the state of these astrocytes? They are reactive. They are already activated. They are already in a state of flux, primed for change. This is the brilliant insight. The problem is also the solution. The reactive astrocytes that form the scar are the perfect candidates for transformation. They are already in the right place at the right time. They are already on high alert. This reactive state, which makes them build the scar, also seems to make them more receptive to being reprogrammed. It's as if their cellular machinery is already warmed up and running, making it easier to steer them in a new direction. So instead of trying to fight the scar, the new strategy is to co-opt it, to hijack the brain's own flawed healing process and turn it into a truly regenerative one. The very cells that rush in to form the barrier, the cells that block recovery, could be instructed to become the recovery itself. They could be told to stop being bricklayers and to start being architects and builders, transforming the wall of scar tissue from the inside out into a new neighborhood of functional neurons. This concept of the glial scar as a double-edged sword is a beautiful example of how science evolves. We move from seeing something as purely bad to understanding its nuances, its dual nature. The brain's injury response isn't a mistake. It's an ancient, evolved mechanism for survival. But with modern science, we might be able to gently guide that response away from simple scarring and towards active reconstruction. The astrocytes, once seen as the builders of the wall, could become the builders of the bridge to recovery. Let's now imagine how this could work in a real-world scenario. Let's consider one of the most common and devastating forms of acute brain injury: a stroke. A stroke happens when a blood vessel in the brain is either blocked by a clot or bursts. In either case, a part of the brain is suddenly starved of oxygen and nutrients. Neurons in that area, being incredibly sensitive, begin to die within minutes. This can lead to the sudden loss of function, the ability to speak, to see, to move a limb. For a long time, the main treatments for stroke have focused on the immediate aftermath. Doctors try to dissolve the clot or stop the bleeding. They try to save the neurons that are in the danger zone, but not yet dead. This is crucial, but it doesn't do anything for the neurons that have already been lost. Once they are gone, the function they controlled is gone with them. The patient is then left with a long, difficult road of rehabilitation, trying to teach other parts of the brain to compensate for the loss. It's about working around the damage, not fixing it. But now, let's introduce our new therapeutic strategy. Imagine a patient is brought to the hospital after a stroke. The immediate damage has been done. A core of neurons in, let's say, the motor cortex, the part of the brain that controls movement, has died. In the hours and days that follow, the astrocytes in that region become reactive. They start to form the glial scar. This is the window of opportunity. At this point, doctors could use a highly precise, minimally invasive procedure to deliver our therapeutic package directly to the site of the injury. Remember our delivery system? The harmless viral envelope carrying the genetic instructions for a transcription factor like Neurobiology. This package would be injected into the damaged brain tissue. The viral envelopes would then infect the cells in that area, with a preference for the reactive astrocytes. Once inside an astrocyte, the envelope releases its cargo. The Neurobiology gene gets to work, producing the Neurobiology protein. This protein, our new librarian, travels to the astrocyte's nucleus. It closes the astrocyte instruction book and opens the neuron instruction book. And then the magic begins. Right there, in the middle of the stroke-damaged tissue, the reactive astrocytes start their transformation. The very cells that were forming the glial scar now begin to morph into new baby neurons. Over the course of weeks, they mature. They grow axons and dendrites. They start to look and act like the motor neurons that were lost. The hope is that these newborn neurons, being in the right place, will be guided by the surrounding environment to become the right type of neuron. They will begin to connect with the surviving neurons on the edge of the injury, and they will try to re-establish the broken circuits. They will reach out and connect to the pathways that lead down to the spinal cord, the pathways that carry the commands to move an arm or a leg. This isn't just a fantasy. This has been tested in animal models with incredibly promising results. In studies on mice and rats that have had strokes, this in vivo cell conversion therapy has been shown to work. Scientists have observed the astrocytes turning into new neurons. And more importantly, they have seen functional recovery. Mice that had lost the use of a paw after a stroke were able to regain movement after the treatment. They could once again grasp and use their paw because the new neurons had helped to rebuild the damaged motor circuit. This is a paradigm shift. We are moving from a strategy of damage limitation to a strategy of active repair. We are not just trying to protect what's left. We are trying to replace what was lost. The therapy isn't some generic pill. It's a hyperlocal targeted approach that uses the brain's own cells in the very location they are needed to rebuild from within. It's like having a team of microscopic construction workers on standby inside your own head, ready to be deployed to any disaster site to begin repairs. The potential for treating acute injuries like stroke or traumatic brain injury is immense. It offers a kind of hope that was simply unimaginable just a generation ago. The promise of this technology extends far beyond sudden, acute injuries. It also shines a light of hope into the dark world of chronic, progressive neurodegenerative diseases. These are illnesses like Alzheimer's disease and Parkinson's disease, which are not caused by a single event, but by a slow, relentless, and heartbreaking loss of specific types of neurons over many years. Let's first consider Alzheimer's disease. This is a disease that robs people of their most precious possession, their memories, their sense of self. At its core, Alzheimer's involves the progressive death of neurons, particularly in areas of the brain that are crucial for memory and thinking, like the hippocampus and the cerebral cortex. As these neurons die, the brain's networks begin to fail, leading to memory loss, confusion, and cognitive decline. Current treatments can help manage symptoms for a time, but they don't stop or reverse the underlying neuron loss. The disease continues its slow march. Now, let's apply our new strategy. The theory is that we could use the same astrocyte to neuron conversion technology to replenish the dying neurons in the Alzheimer's brain. The idea would be to deliver the transforming transcription factors to the hippocampus, for example. The local astrocytes, which are also in a reactive state in the Alzheimer's brain, could then be converted into new hippocampal neurons, the very type of cell that is being lost. In theory, this could help to rebuild the failing memory circuits, potentially restoring lost cognitive function, or at least halting the decline. Similarly, let's think about Parkinson's disease. Parkinson's is primarily a movement disorder. It causes tremors, stiffness, and difficulty with walking and balance. The root cause is the death of a very specific type of neuron in a small area of the midbrain called substantia nigra. These are the neurons that produce dopamine, a crucial neurotransmitter for controlling smooth, purposeful movement. As these dopamine-producing neurons die off, the brain's motor control system starts to break down. The therapeutic goal here would be incredibly specific, to create new dopamine-producing neurons right there in the substantia nigra. The strategy would be to deliver a cocktail of transcription factors that don't just say, become a neuron, but that say, become a dopamine-producing neuron of the substantia nigra. If the local astrocytes could be coaxed into this very specific transformation, they could start producing the dopamine that the brain is missing. This could potentially reverse the motor symptoms of the disease in a way that current dopamine replacement drugs cannot. It would be a true biological repair, not just a chemical patch. However, treating these chronic diseases presents a whole new level of complexity compared to an acute injury like a stroke. The challenge is immense. In a stroke, you have a one-time event. You can go in after the fact and try to repair the localized damage. But in Alzheimer's or Parkinson's, the disease process is ongoing. The brain environment is toxic. There are sticky plaques and tangles in Alzheimer's, and other pathological processes in Parkinson's. These are the very things that are killing the original neurons. So, a major question is, if you create new neurons, will they just be killed off by the same hostile environment? It's like trying to plant new trees in a forest while a fire is still smoldering all around them. The new trees might not survive. For this therapy to be successful in the long run for neurodegenerative diseases, it would likely need to be combined with other treatments that address the root cause of the disease. Treatments that clear the plaques, reduce the inflammation, and make the brain environment healthy and welcoming for the newly born neurons. Furthermore, the brain in a long-term degenerative disease undergoes much more widespread changes. The circuits are not just broken, they are slowly and progressively dismantled over years. Integrating a new neuron into a circuit that has been degrading for a decade is a much bigger challenge than integrating one into a circuit that was healthy just a few days ago. Despite these significant hurdles, the potential is too great to ignore. This research has opened up a completely new avenue of attack against diseases that have, for so long, seemed unstoppable. It represents a shift from a defensive posture to an offensive one. Instead of just trying to slow down the destruction, scientists are now seriously pursuing the possibility of reconstruction. It offers a tangible biological basis for hope. The idea of curing a disease like Alzheimer's may still be far off, but the idea of repairing the damage it causes has, for the first time, become a scientifically plausible goal. The journey will be long and difficult, but for the millions of people affected by these diseases, this new science represents a profound and powerful reason to be optimistic about the future. While the promise of turning astrocytes into neurons is dazzlingly bright, it is important to walk with our feet on the ground. The path from a groundbreaking discovery in a laboratory to a safe and effective treatment in a hospital is a long and winding one, filled with challenges and hurdles that need to be overcome. The scientists working in this field are not just dreamers. They are meticulous problem solvers, and they are keenly aware of the obstacles that lie ahead. Let's gently explore some of these challenges, not to diminish the hope, but to appreciate the true complexity and elegance of the brain they are trying to repair. First, there is the challenge of efficiency. In the early experiments, the conversion rate from astrocyte to neuron was often very low. You might introduce the transforming factors to 1,000 astrocytes, and only a few dozen would successfully make the complete journey to becoming a functional neuron. While this is improving all the time with new and better transcription factors, achieving a high enough efficiency to meaningfully repair a large area of damage is a major goal. It's like trying to reforest a whole valley by scattering seeds from an airplane. Some will land in fertile soil and sprout, but many will not. Scientists are working to understand what makes some astrocytes more receptive than others, and how to prepare the soil of the brain to make the conversion more likely to succeed. Second, and this is a huge one, is the challenge of neuron subtype. The brain isn't just made of generic neurons. It is a metropolis populated by hundreds, perhaps thousands, of different types of neurons, each with a specific job, a specific shape, and a specific set of connections. There are excitatory neurons that rev up the network, and inhibitory neurons that calm it down. There are the huge pyramidal neurons of the cortex, and the tiny granule cells of the cerebellum. A new neuron is only useful if it is the right type of neuron for that specific location and function. You can't just replace a lost violinist in an orchestra with a drummer and expect the symphony to sound right. You need another violinist. So the challenge is not just to turn an astrocyte into a neuron, but to turn it into the correct subtype of neuron. This requires a much more sophisticated recipe. Scientists are now experimenting with combinations of different transcription factors, trying to find the precise molecular recipe that instructs an astrocyte to become, for example, a dopamine-producing neuron for Parkinson's, or a motor neuron for a spinal cord injury. It's moving from a single instruction, become a neuron, to a detailed blueprint. Third, there is the critical challenge of functional integration. This is perhaps the most complex puzzle of all. A new neuron, even if it's the perfect subtype, is completely useless if it doesn't wire itself correctly into the existing neural network. Building a new house in a neighborhood is one thing. Connecting its plumbing, electricity, and internet to the city's grid is another thing entirely. The new neuron needs to grow its axon to the right downstream targets, and it needs to form synapses with the right partners. It also needs to have the correct receptors on its dendrites to receive the right signals from its upstream neighbors. If a new neuron integrates incorrectly, it could be ineffective at best and harmful at worst. It could create noise in the system, or even generate aberrant signals that could lead to problems like seizures. The brain's developmental wiring process is a symphony of incredible complexity, guided by a host of chemical cues. The hope is that the adult brain, even after injury, retains some of these guidance cues, and that a newborn neuron will be able to listen to them and find its proper place in the circuit. Proving that these new neurons integrate properly and contribute positively to the circuit's function is a key focus of current research. It's about ensuring the new musician not only plays the right instrument, but also reads the sheet music and listens to the conductor. Finally, there is a challenge that comes from the very nature of the process itself. When you convert an astrocyte into a neuron, you gain a neuron, but you also lose an astrocyte. We began this journey by talking about how important astrocytes are. They are the gardeners, the janitors, and the security guards of the brain. They maintain the delicate chemical balance. They feed the neurons. They protect the brain. So, what happens to the local brain environment when you take away a significant number of its support cells? Could converting too many astrocytes in one area lead to unintended side effects? Could the remaining neurons suffer because their support staff has been depleted? This is a serious consideration. It's a delicate balancing act. The goal is not to eliminate astrocytes, but to repurpose a fraction of them in a targeted way. Some scientists are exploring the idea that other nearby astrocytes might be able to compensate to take on the extra workload. Others are investigating if it's possible to encourage the remaining astrocytes to divide and replace the ones that were converted. It's a reminder that the brain is a complex ecosystem. You can't change one part of it without considering the effects on the whole. These challenges are not roadblocks. They are the research questions that are driving the field forward. They are the puzzles that brilliant scientists around the world, from diverse backgrounds and institutions, are working to solve. Each one of these hurdles is a frontier of neuroscience in itself. Overcoming them will require ingenuity, collaboration, and a deep and abiding respect for the complexity of the brain. The journey is far from over, but the destination, a future where we can truly heal the brain from within, is a powerful motivation to keep moving forward. We began our journey tonight with a story of permanence, with the harsh decree of Santiago Ramon y Cajal. He gave us a picture of the brain as a magnificent but fixed and immutable structure, a masterpiece that, once damaged, could never be made whole again. For over 100 years, this idea has shaped our view of ourselves. It has been a source of awe for the brain's complexity, but also a source of fear for its fragility. It has defined the limits of medicine and the boundaries of hope for those with brain injuries and diseases. But tonight, we have explored a new story, a story of hidden potential, of cellular shapeshifting, of regeneration where we thought none was possible. This research into the transformation of astrocytes into neurons doesn't erase Cajal's legacy. It builds upon it. Cajal described the world he could see with the tools he had, and he described it with genius. But science is a continuous journey of looking deeper, of developing new tools that reveal new layers of reality. And this new layer has fundamentally reshaped our understanding of the brain's nature. The central dogma is no longer that the brain is static. The new understanding is that the brain, even the adult brain, possesses a remarkable and previously hidden potential for self-repair. It is not a fixed, hardwired machine like a computer, where a broken transistor is broken forever. It is much more like a dynamic and resilient ecosystem, like a garden or a forest. In this garden, even after a fire has swept through and left a clearing, there are seeds lying dormant in the soil. These are the astrocytes. And with the right catalyst, the right molecular signal, these seeds can be coaxed to sprout, to grow into new trees, and to begin the slow, patient work of reforesting the damaged landscape. This shift in perspective is profound. It changes the entire paradigm of treating brain disorders. The old paradigm was defensive. It was about protection and compensation. Let's protect the neurons that are still alive. Let's try to train other parts of the brain to take over the lost function. These are still valid and important strategies. But the new paradigm is proactive. It is about reconstruction and replacement. It asks a much bolder question. Can we replace the very neurons that have been lost? This changes how we see the brain itself. It is not a fragile artifact to be kept behind glass. It is a living, breathing, and surprisingly plastic tissue. The word plasticity in neuroscience usually refers to the brain's ability to change the connections between neurons. But this astrocyte conversion reveals a much deeper form of plasticity, a cellular plasticity. The very identity of the cells themselves is not fixed. The brain contains within it the cellular seeds for its own potential reconstruction. This discovery fills us with a sense of wonder. It reveals the elegance and economy of nature. The solution to brain repair might not be some artificial cell we have to invent and transplant. The solution may have been there all along, hiding in the most abundant cell in the brain, waiting for us to discover the key to unlock its potential. It is a story of hope, not just for medicine, but for our appreciation of the resilience of life itself. The post-mortem discovery, in particular, leaves us in a state of quiet contemplation. It blurs the lines we thought were so clear. It suggests that potential, that a latent spark of biological possibility, can persist even in the stillness after life. It's a humbling thought that reminds us how much we still have to learn about the fundamental nature of living systems. So as you lie here, ready to drift off to sleep, let this new image of your brain settle in your mind. Do not think of it as a fragile, finite machine. Think of it as a vast, enchanted forest, a dynamic, living city of stars, a place of immense complexity, yes, but also of deep and profound resilience. A place where the gardeners hold a secret identity, where the support staff has the hidden potential to become the architects of their own renewal. The journey from this beautiful scientific discovery to a widespread cure is still long. But the path is now lit. The hope is no longer just a dream. It is a tangible scientific reality being built, piece by piece, in laboratories around the world. And it all started with looking at a humble star-shaped cell and asking a simple question. What if you could be something more? The brain is not set in stone. It is a garden. It is a city of light. And it holds the secrets to its own healing. Quietly, patiently, waiting, sleep well.